geneticist at the forefront of precision medicine. He holds the Canada Research Chair in Translational Genomics and uses genome sequencing to understand causes of cancer, guide treatment of patients, and detect early disease using blood tests. Dr. Pugh is a professor in the University of Toronto Department of Medical Biophysics, a senior scientist at Princess Margaret Cancer Centre, and a senior investigator at the OICR, so the Ontario Institute for Cancer Research. And he's director of the OICR Genomics Program and the Princess Margaret Genomics Centre, genomic cores that specialize in single-cell, cell-free DNA, and clinically accredited whole genome sequencing. So Dr. Pugh's been recognized by Canada's top 40 under 40, the Canadian Cancer Society Bernard and Francine Dorval Prize, and inducted into the Royal Society of Canada's College of New Scholar, Artists, and Scientists. So that's a very long and impressive bio. So no, that's great. Thank you. So we're really looking forward to your talk.
Okay, great. So today I'm going to talk about how we use genome technologies to measure the cancer system and the immune system. And it turns out patients with cancer have this ongoing battle between cancer cells and immune cells. And the latest revolution in cancer treatment in general have been these immunotherapies that activate those immune cells and give them an advantage to clear out those cancer cells. So I'm going to be talking a lot about genome sequencing, exome sequencing, some cell-free DNA work that we've done, and then finish with the newest technology that we've been working on, which is a T-cell repertoire sequencing analysis approach. It's probably the technology I'm most interested in because it really makes use of extraordinarily large data to try to make sense of very complex immune responses. So this is called Inspired by Immunotherapy. Inspire is a inside pun in that the clinical trial I'm going to talk about is an acronym that spells out Inspire. But yeah, it's all about measurement, cancer, and immune systems. Some disclosures. I guess the main one is we've patented the TCR sequencing approach. So there's a conflict there. The whole crux of everything that goes on in my lab is really about knowing that tumors aren't just cancer cells. They have cancer cells in them, but they've got a huge diversity of other cancer cell types. And they're not just immune cells, there's stromal cells, there's blood vessel invasion. And now we have genome technologies to infer the presence of these cells. And not in this talk, but definitely where the field is now we have single cell technologies that let us pick out and measure the RNA from individual cells. This work was done right before that revolution, so it's mostly going to be based on grinding up these tumors and then inferring the presence of alterations in all these cell types. I mentioned we're going to talk about a clinical trial. The trial we're going to talk about is using immune checkpoint blockade. So what normally happens, the way to read this plot, the top is a T cell, bottom is a cancer cell, and T cells turn on this PD-1 reaction here. So essentially, this is a reaction that says, don't eat me, and shuts down the immune cell, and basically allows cancer cells to evade the immune system. And therapies like the one we're going to talk about is called anti-PD-1. There's also anti-PD-L1. These are basically therapies that disrupt that interaction. They reactivate the T cells, and they can eat or attack the tumor cells. So I mentioned Inspire. That's the acronym up there. Inspire was a clinical trial of immune checkpoint blockade, pembrolizumab, but the real secret sauce there was very biomarker and biospecimen rich. So every patient gave a biopsy right at enrollment in the trial. They also had another biopsy very soon after, so 3 cycles of treatment later. Really great opportunity to look for changes in cancer system and immune system. And it had a huge amount of blood collection. So that's what those syringes are. They obsessively collect blood every cycle. So we really look at some of those dynamics non-invasively using cell-free DNA. I'm not going to talk about it today, but there's a big imaging cohort as well. So if you're interested in imaging, it's a good chance to plug into that data set. The other novel piece was the cancer types. So immune checkpoint blockade had been used almost primarily in lung, or exclusively in lung and melanoma. At that point, all those tumors are bristling with mutations. The idea was they're very immunogenic. We went exactly the opposite direction. So there are 5 cohorts that hadn't been tried for pembrolizumab before. So head and neck, breast, ovarian. Melanoma was kind of a positive control. Those patients did great. And then mixed solid tumors, which turned out to be a grab bag of mostly sarcomas. Yeah, and led by Lillian Su, a medical oncologist, and Pamela Haushi, an immunologist. Yeah, please. Oh, yeah, and please break in. I think this is more of a workshop environment, so happy to take questions.
Just to understand, so this is, so syringes are blood samples and the treatment is something, or
The
you,
treatment is every arrow. So this is a
Every
drug that's
arrow
given
is a
roughly every
treatment
10
thing?
days.
I
So
see.
you get recurrent.
Oh, then the sampling of blood is much more. Okay, so
So the
syringes
patients are
actually
coming in
treatment.
anyway. They get the drug and you get the blood
Everything in the same
simultaneously.
thing. And this is
And then these little
chemo,
clumps are supposed to be tumors.
and this is the PD-1 blocker. basically.
Exactly.
That's what you get.
Yeah.
Okay, got it.
Yeah.
Yeah. Hello. Was there any control in this experiment? I mean, like tumors that are not treated by this treatment, at least, but treated by another one, just to check what's happening with the composition of tumors?
Yeah, not at all.
In
So it
absence
was
of that,
the,
just a baseline.
the control is longitudinal rather than a two-arm study. So it's a single-arm study, and the control is to compare biopsies serially within each patient. But we don't have a matched untreated arm.
Okay.
Yeah, it would be nice, but difficult to arrange. I also mentioned for each biopsy, exome, RNA-seq, immune profiling, and TCR sequencing. So the first challenge was these biopsies are really small. They're often little core needle biopsies. You almost think of them like a little piece of pencil lead. So they're really, really tiny. So the first challenge was to do as much as we could with these really small samples. So we got the blood samples. Those were plentiful. Those are 30 to 40 mils of blood. The tumors were all digested and pooled. So this is already a challenge. We lose the spatial information. Some of the biopsies are mostly immune cells. Some of them are mostly tumor cells. We've digested and pooled them all together. And then we use flow cytometry to isolate. So on average, we got 250,000 cells. We use flow cytometry to isolate cells and then hand them out to all these labs. So luckily for my group, we were at the top of the list. We always got exome and RNA-seq on basically every single patient. And then we kept trying to go further and further down the list. If we were really lucky, we got all the way down to patient-derived xenografts. Those went to Lori Ailes' group. There's some immune phenotyping panels. And then right at the end, I don't think we ever got here. If there's really leftover tumor-infiltrating lymphocytes, those could be expanded for our research on TILs. And then we did exome and RNA sequencing. The first take-home lesson is how important it is not to use targeted approaches in genome science. At the time, we used exome sequencing. That's what we could afford. All of our projects now are whole genome sequencing, which is what this slide refers to, because you get all manner of cancer genome variation, the smallest change, point mutations, you get big cytogenetics changes. The RNA tells you a lot about splice isoforms and genes maybe you didn't think to look at when you started, but became really important later on. So if you're embarking on a genomic study today, I definitely recommend just jump straight to exomes and RNA-seq. Don't let the price tag pay you off or scare you off, because that type of data really ages very well. And we just kept going back to this data set with new questions year after year. The first challenge, maybe related to your point about the lack of a control, is the cohort turned out to be super heterogeneous. It's 100 patients, so they're colored by the 5 cohorts. They kind of had variable responses. So the melanomas did, ultra well. So the way to read this is the change in tumor measurement over time. So you want to be, this is a waterfall plot, you want to be down on this plot. So melanomas did really well. Triple negative breast tumors did not do well, except for this one patient who was an extremely strong responder. That's one of the worst breast cancers you can get. She did amazing. And this was kind of the point of the study. We wanted to gather together all of the responders and non-responders and look for some of these shared biomarkers across cancer types. And you can kind of see these waves here that kind of map the patients to progressive disease, stable disease, partial and complete response, and not evaluable. So it was really tough just looking at the genomes. They were like full of weird mutations. They were really driven by cancer type. So we couldn't really make sense of the data in the moment, just using imaging-based response. But we did have a huge number of serial blood plasmas. And I showed this slide just to show how complicated it is to collect a blood sample from a patient. And we had to do this 300 times because we wanted to get at least 3 blood samples from 100 patients. The way this works at Princess Margaret now is the patient consents, and there's a whole documentation of consent arc in blue. Then the other secret to getting this to work is all the patient, when they come in for their clinical bloods, we collected their research bloods simultaneously. So it was just one poke with the needle. but they'd get their tube for clinic, but then it could also participate in research very easily. Really important, especially for these longitudinal studies. And then we had porters who were able to grab all the blood as soon as it was drawn and then delivered to our biobank. So it's kind of what that orange arc is all about. I've been involved in a few studies where it seemed very easy to get the blood samples or to write into the grant for getting blood samples, but there's actually a huge piece of machinery that has to be in place to actually collect those blood samples reliably over time. So since we had done exome sequencing, we had lists of mutations. Even though they're kind of scattershot all over the exome, we did have lists. And at that time, there was a then new company called Natera, and they had a new technology that relied on knowledge of mutations in the genome. So that was very important measure. First important measurement, number one, was just lists of mutations. So what this group does is they take your whole exome sequencing data and they'll pick out the 60 mutations that they think are easiest to find. The algorithm is proprietary, but it seemed pretty obvious they were choosing mutations that are in copy number gains. So each cancer cell could give you 4 or 5 copies of the mutation. And they always chose clonal mutations. They weren't going after subclones. So we gave them lists of mutations, and their algorithm basically chooses the easiest of 16 most easy to find. And then they generate a bespoke assay special for that patient. And once they have that assay, they can test as many blood samples as you want. So it's very effective for looking at treatment after diagnosis. So their goal isn't early detection, it's get the tumor sequence and follow it in a very sensitive way over time. And then you get plots like this in spot number 5. You get treated, and then you expect to see either a decrease or an increase over time. So this is a brand new assay at the time, but it really gave us a path to give us a more precise molecular measurement of response rather than relying on imaging. And the first finding there was just the,
Sorry, quick one. The panel's fixed throughout, right?
It's fixed
For
per
each patient. Okay.
So you can't change it for the patients. You can't catch relapse. You can't catch new mutations. You just know it, you know what diagnosis, and you follow it.
Okay, thank you.
Yeah. A really important question because it totally changes your design if you're looking for relapse, if you're looking for early detection. But it's very good for this very specific application. Yeah.
But wouldn't you see relapse? I mean, it could be in the background of one of the things. So wouldn't it go?
Oh,
up?
it's for sure there in the blood. But the problem is their test is so focused, they only see the 60 mutations they set out to see.
But sorry, what I don't understand is why should all 60 of them decrease?
They might not. If it, oh yes, if there
That's what I
is
mean.
relapse, all 16 will come up over time.
Oh, they all linked.
That's
They're
true.
all linked. They're all fixed.
Yeah, you
Got
just
it.
can't find
There's no
new
differences.
mutations.
Got it. Yeah,
Yeah.
new I understand, but you're saying even differences is not there because these are the ones which are all fixed.
Right,
Either they all go
because
up or
they've
all get down.
yeah, because they've
Got it.
chosen the clonal mutations that are shared across presumably every cancer cell. We actually, I don't remember if I have a slide on this. We actually know they didn't get it right 100% of the time. Sometimes one mutation would drop off, but the other 15 would keep going. So we basically said you had, I think 5 of 16 was like the number to say you had disease versus not. So this was the first finding. So this is the swimmer's plot showing survival, and we sorted every patient benchmarked to baseline. So the other surprising finding at the time, maybe not surprising, each patient had a different starting level of tumor burden. So some were very high, some were very low. So we took that day 1 untreated time point as we normalized them to 0. Then we asked at the next time point, was there an increase or a decrease? So that delta is what is marked in red and blue. So patients who decreased are the ones that were doing better, and the patients who with the increase in cell-free DNA were the ones who were surviving much, much shorter. So if you look at the lengths of the swimmer's plot, we did this more formally, you can see the patients on the bottom, a lot of X's, those are the patients passing away, so their swimmer's plots are lower, whereas the patients with the longer cell-free DNA don't have as many patients passing away, and their swimmer lanes are longer. And here's the data summarized over here. So the patients by imaging with complete or partial response, they went down and they stayed down, except for this one outlier. And then this patient here, these patients here with stable disease, all of them started at 0 and they all went up. So just looking at the change, kind of a gross metric right at the start, was a really effective way to stratify, to predict responders from non-responders extremely early in treatment, like right from their first blood draw. And this signal really held up long term. So just looking at Kaplan-Meier plot, just looking at increase on cycle 3 versus decrease in cycle 3, you could basically predict overall survival, but also progression-free survival. And it really held very nicely across all the cohorts. So this was also a case of taking a clinically kind of a messy cohort, these 5 different disease types, applying one genomic tool across all of them. But every single cohort really showed this pattern, really in a tumor agnostic way. The other and most powerful predictor of response was who went from disease detected to flat 0. So we called this clearance. So clearance here is 0 of the 60 mutations were detectable at cycle 3. And those are the patients who we rarely use cure in cancer, especially cancer clinical trials, but I think this is about as close to it. And actually, a lot of these patients who went to 0, wasn't a huge number of them, but they're the ones who continue to cruise to this day. They really had these really dramatic, very, very early responses. And if you actually change the paradigm from change in cell-free DNA binned by imaging to instead clearance versus non-clearance, you can see these patients are the ones who cleared and then just kept going, except for this patient here who had a very strong relapse. But in general, this is held up as a very strong biomarker across immunotherapy now. That very early blood draw is a very, very strong predictor of how you're going to do. And all 12 patients with ctDNA clearance were actually alive after 25 months median follow-up, apart from that one with the increase in signal. Virtually all of them are still alive today. They've just kept going extremely robustly. They've had these long-term survivals. The other interesting finding is over on the right is the change in cell-free DNA vastly predated the change in tumor shrinkage as measured by imaging. So just 2 examples here in this melanoma case. In black is the cell-free DNA level, just crashed to 0. And then slowly that melanoma is kind of melting away. But it really gave you this look into the future by using that early blood test. And same sort of story in head and neck, that like really dramatic crash. And then the tumor as measured by CT slowly going away as well. So really showing that using this blood test gave us that direct look straight into the cancer system as it responded. So we had all this blood work, we published that. Natera actually went on and got a Medicaid filing based on this study. I couldn't believe it. So they did very well financially. We got a paper out of it. But we also got a tool to better stratify our patients. So we had these very messy genomes, but now we didn't have to rely on imaging-based response. We could now instead reimagine response on 2 axes, a change in cell-free DNA and a change in tumor measurement by imaging. So the idea is not just throw imaging out. It's super important. It's a vital diagnostic tool, but cell-free DNA helps you address some of these nuance. And we invented these 4 new groups. I'll start here. It's the easiest one, high sensitivity. Those are the patients' tumor shrank, cell-free DNA shrank. They're responders. Also on the up right-hand side, low sensitivity. They're basically totally resistant to immune checkpoint blockade. Tumor grew and cell-free DNA grew. Also makes sense. The 2 interesting cohorts are these ones here. So this one, mixed sensitivity with emerging resistance. So the tumor is shrinking, but the cell-free DNA is going up. So it shrank a little bit, but there's still a resistant population there that gives rise to relapse. And then mixed sensitivity with potential pseudoprogression. This is the most interesting to me. Cell-free DNA dropped, but the tumors are growing by a huge amount. This is the log scale here. And this is actually due most often to, since you're using an immune activating agent, the tumors actually swell. They like get invaded by these immune cells. Radiographically, it's like, whoa, these tumors are out of control. But there's no cell-free DNA there because the cell-free DNA is, the cancer cells aren't there, they're being wiped out, and the immune cells are the ones that are combating them. So this is probably the most nuanced contribution because you're actually able to differentiate these 2 different changes in size by imaging.
Sorry, is that's best % change?
Pardon?
That's the best % change in tumor mass?
That's right.
Thanks.
Yeah.
Okay, so it was.
So are you saying that the tumor volume is actually not a good variable?
You can't say it's not good
Because you
in
got
most
big
patients,
pictures.
in most patients it's pretty good. But in these edge cases, especially in the context of immunotherapy, you need more. You need an orthogonal method using cell-free DNA as
Right.
well.
So, sorry, what I meant to say is that if you get some progression with cell-free DNA, you would trust that even if your tumor volume was going not in the right
Yeah,
way.
I think that's the big question. So that's
Okay,
where the
that's amazing.
prospective studies really come in and actually test, especially the clinical utility. Our clinical teams, when you get a cell-free DNA result, willing to act purely on that. We're doing those studies now. I don't know, the jury's still out a little bit. It kind of depends on which platform you're using. Imaging is very hard to leave, I think.
Could I just, sorry, just to go back to the previous slide, but I think it's actually recapitulated here too as well. The proceeding of the drop or increase or mix, I guess.
Yeah.
Is there consistency in this time between that you
No,
see?
there's not. It's very irritating. I kind of thought it would be like crash and then you'd be like, oh, I always got 6 months lead time, but sometimes not. Sometimes it's like a year. Sometimes they're never overlapping. Cell-free DNA is always a little bit early, but sometimes they're pretty close.
And does that depend on tumor size at that point? Like, could it be the number of cells in some
No, I
respects?
can't say that larger tumors melt faster. I don't think that's the case, which is kind of what I thought going in, but no. It's confounded because the larger tumors do give huge amounts of cell-free DNA. So your baseline start is also much higher. So this isn't a huge number of patients. It's kind of tricky to tease out. So we took our new groups and we redid the survival analysis, and it kind of showed what you'd probably expect. High sensitivity doing really well, low sensitivity not doing well. But then we also had these two middling groups that kind of separated out that gray area. So the difference between red and blue is now much more significant because they're not kind of contaminated by the two middling groups. But the two middling groups kind of make sense. The one piece I'm still bewildered is the flip between this yellow and blue line, and then the yellow and blue line over here. So pseudoprogression related to overall survival, but not progression-free survival. And I think it's because of how we define progression-free survival, which is often based on imaging. So you can easily tie yourself up in that a little bit trying to unravel how this model then maps onto how we measure survival versus overall survival. What I really want to do is take these four clean groups and apply that in a new cohort where we can say, this is how we're defining response, and then do all the correlates based on that. Whereas here, we still have to, we kind of inherit the traditional measurement of survival and response, which is imaging. So just to explain that funny little swap of lines. Okay, so I mentioned before we did a bunch of genome sequencing. We got a huge number of drivers. This is microscopically small because we found like tons of drivers and they were scattered all over the place. So even trying to, we took our new groups and we put them into high sensitivity, clinical benefit, and low sensitivity. And the point is not that there was one magic mutation that beautifully segregated, except for the BRCA2 mutations. And this was actually published almost around the same time we did, where they found if you have a BRCA2 mutation in your tumor, it's a very good sign. It actually turns out to be an immunogenic subtype of cancer. And you can see that's the only mutation that's fully exclusive in the high sensitivity clinical benefit group. All the other ones are kind of mixed across. And really what our lesson here is in the lower right is how important it is to do personalized testing really for each patient. It's very hard to say there's one magic signature or one panel. But really how important it was to cast as broad a net as possible for the genomic analysis, because really every patient had their own mechanism specifically to evade the immune system.
I can read the question.
Oh, good.
Sorry.
Yeah,
Yeah. So the
sorry.
question is
Oh,
it
good.
possible that the swap of lines could be due to progression in a different clone of cancer cells not detected by the cell-free DNA?
I think that's unlikely because we chose the clonal mutations. So it would have to be a subclone that didn't inherit any of the clonal mutations, which I could dream up a way that could happen. It could delete them all, but all 16 of them, that seems very unlikely. And part of the algorithm is they don't put probes on the same chromosome. So you'd have to have 16 chromosomes all deleting at the same time. So I don't think that's the reason. Okay, so we did want to make sense of this data. What in here actually is associated with response? So the first one was tumor mutation burden and expression of PD-L1. These are already biomarkers kind of established in the field. They were statistically significant. They were correlated with response. It's what held up with other studies in lung and melanoma, but they weren't perfect. So every time looking at tumor mutation burden here, the hypothesis going in is you have more mutations, therefore you're expressing more mutant peptides that the immune system can identify as foreign. Therefore, they should be more easily detected, and therefore you'd respond. But to my eye, it's not very satisfying because there's lots of low mutators that are not responding, and there are lots of high mutators who are responding. So it's really an imperfect biomarker. And it's actually a biomarker that's kind of fallen off a little bit, really effective in lung and melanoma. But when you go into our 5 cohorts, which are kind of medium mutation rate, I wouldn't say that you don't want to hang your entire hat purely on tumor mutation burden.
Sorry.
And same thing with PD-L1. Yeah.
Sorry, yeah, I no offense. Since that bothers me, it's you have 5 different cancer type, and of course the melanoma going to have high TMB, and that would dominate all of the results. So I just wondering that do you want to separate them and look at the no mutation one, but they also respond? So
yeah, and we did split them out. This signal is not driven purely by melanoma. We had a couple of very high mutation rate from the other cancer cohorts, but yeah, it's absolutely an issue. I think it's more a biomarker of immunogenicity, but I actually think the immune system is more likely to recognize the wild type antigens that are being expressed in the wrong context, like testis antigens being expressed in breast cancer. That the immune system is going to stamp those out right away. I think I have no data to back this up, but I think that's the more likely reason for immune response in these tumors.
yeah, thank you, I agree.
I talked about BRCA, so we dipped into another data set published by Memorial Sloan Kettering, the IMPACT cohort. They've done an amazing job just making all their data public for anyone to use. It's very easy to make this Kaplan-Meier. I think this figure took under a minute. You just can go to this great CBio portal system, search for your favorite cancer type, sort by mutation. They give you the survival analysis. So it really held up nicely in our hands, but not associated with PD-L1 score. PD-L1 expression by protein immunohistochemistry has also kind of fallen off as a biomarker. The antibodies aren't very good. How they get scored is quite heterogeneous. So there really has been a move really away from using PD-L1 exclusively and moving more to genomic and cell-free DNA biomarkers since we did this work. The other big finding was loss of B2M. So B2M is part of the protein complex that presents these antigens to the T cell. So there's which one's the T? Yeah, the T cells down here at the bottom. This is the basically review of all the ways that the cancer genome can use to evade the immune system. And one of them is delete B2M. Therefore, it can't present antigens and becomes invisible to T cells. We only ever saw this deletion in patients who were non-responders, and we never saw them in the high sensitivity case. So really example, a very strong biomarker with a good mechanistic understanding. If you can't present your antigenic peptides, the immune system's not gonna be able to find you. So a very handy, easy to measure biomarker, especially from exome sequencing, because that whole chunk of DNA is just dropped right out. Since we're now in the tumor world, we still wanted to learn everything we could from cell-free DNA. We knew how important the delta was. So instead of looking at delta cell-free DNA, we looked at delta gene expression. So we're looking at the baseline and that first biopsy. And you can see the patients who are high sensitivity have tons of genes all being upregulated. So these are movable cancers. You treat them with PD-1 and that tumor is changing. That's the whole point of therapy. Whereas you can see the low sensitivities, very few differentially expressed genes. They're like these rocks. You try to treat them and you just can't move them. And of course, the question is, what are these genes? They're all being massively upregulated. Probably not surprisingly, it's all about immune activation. So we're looking for genes that get activated. Essentially, immune checkpoint blockade is causing this crashing in of immune cells. And when we do RNA sequencing, we see all those transcripts. Yeah.
Sorry, just is the way of thinking about in the previous slide is just not access to these guys, or like none of your
there are
immune
so
cells
many reasons. Is
there's
it
so
the
many
drug can't get in?
Yeah, whatever.
Is
Yeah,
it
is that
there's
all that the way?
exclusion?
Yeah, or is it really?
Is it, do the immune cells have to, actually way back on my first slide, I showed the immune cells interleaved with the tumors. Some tumors are very good at immune exclusion. We had none of that information. This was all from inference. But based on this data, that's where we've gone now, is starting to do spatial work to look at those types of patterns. But at the time, we just digested them and handed out cells to a bunch of labs. I actually don't know yet whether we have a good answer. I've seen these very striking images of like armies at the gate, but I don't know if we actually see that often. So when we do look at those genes, it's all immunity, lymphocytes, B cells, it's all genes associated with immune response. And then the ones that are all downregulated are all the cancer signals. It's chromosome segregation, proliferating cells. This is a very satisfying analysis because it was so well controlled by having those serial biopsies. It's very hard to do if you have biopsies at 2 time points from different people. But I think going back to the same site twice, being able to say, and they actually went out of their way to get that second biopsy, so you could really do that differential analysis in a more controlled way for that individual patient. The other change using RNA-seq, you can take gene lists associated with that other groups have published and test them out. So at the time, there were 3 signatures: an immune signature, an interferon gamma signaling signature, and a cytolytic activity signature. I put a check mark on the middle one. That's really the only one that held up. This one was pretty good, but it was more of a surrogate for immune cells. And that interferon gamma signaling signature, we've actually looked at in several additional studies, and that's really the one that has held up across multiple cohorts and multiple immunotherapy studies. But again, it's all about the delta in these ones. You see that signature changing over time. And you can basically use almost the same algorithm, but instead of taking lists of signatures, you can take lists that are informed by single cell sequencing experiments. So in parallel groups, it's sorted out naive B cells, memory cells, plasma cells, all manner of T cell populations. And they said these genes are unique to these cell types. And you can take those lists and then go check and see if your tumors are enriched for them. And the tumors that were the ones who were responding were all these CD8 T cells. So CD8 T cells are the killers. They're the most important T cells in the tumors. And treatment with pembrolizumab resulted in a huge increase in the frequency of these CD8 T cells. We also picked out a few other ones, the follicular helper cells, which turn out to be important for these tertiary lymphoid structures that sort of get built inside of these tumors, and they actually become these little immune cell factories. And gamma delta T cells, which at the time we had no idea what they were doing, but the entire gamma delta T cell field has now absolutely exploded. These are very highly specific to cancer antigens, and they're now a hot lead for trying to develop new therapeutics based on these gamma delta biology. So it was kind of an incidental finding at the time, but I haven't harped on it enough how important it is to sequence everything and then test out a bunch of ideas using that data. And that's where the delta gamma finding came from. So we want to get beyond just presence. So I've got a T cell here. There's an antigen presenting cell, which can be a tumor cell. And the way this works is these tumor cells are presenting up these antigens in this MHC complex. Actually, this thing is in the middle. And then the TCR is supposed to find them. And if it recognizes them, go off and mature and then destroy those APCs. So we want to know, is it not just T cell presence, but is there something about the T cell specificity that can help us better understand response to these therapies? So we had this technology. This is where my conflict at the beginning came in. We developed this probe set that basically baits the VDJ rearrangements that are unique to T cells. So the way that this antigen is detected is through these rearrangements of the T cell genome. So the rearrangements look like this. The genome is full of a bunch of V's, a bunch of D's, and a bunch of J's. And the T cells will actually rearrange their genome, delete the V's, D's, and J's they're not using, and bring together these segments that are completely unique. It's a highly unique barcode for a very short 15 to 18 amino acid stretch of protein. So this is a very powerful barcode because you can basically take a whole genome sequencing library. Our technology baits out these sequences, and at the end of the day, gives us a highly enriched library specifically for VDJ sequences. And from those VDJ sequences, we can say this T cell is specific for this antigenic target. Yeah.
This is in biopsy.
So the nice thing with this technology is it's hybrid capture. So it works on fragmented DNA. So it can work on cell-free DNA.
So you can look for, so you see this stuff in cell-free DNA?
So that's why we built it. Yeah, it was actually for the cell-free DNA question, but works in biopsies too. So data looks like this. So if you look at a blood sample, we've got hundreds of thousands, millions of unique T cells in our blood. Each little tiny slice here is unique rearrangement. Contrast that to the far right, a T cell lymphoma that should be almost basically one rearrangement. It's one cell that's hyperproliferated. And you can kind of see that here. It's like one giant clone. You can sequence it to death. You basically just find the same thing. So yeah, I mentioned this method is nice because if you can build a genome library, you can pull out the TCR receptor sequences. And then we have a method to read out the TCRs. And we calculate 3 metrics, a specificity metric. So is it specific for one antigen that we know? Diversity. So do you have a polyclonal, oligoclonal, or monoclonal invasion? Functional state, we've tried to do from our TCR data. That's much better done by single cell RNA sequencing. So we've more stuck to the top 2 metrics. So yeah, we wanted to answer 2 questions. What is the change in diversity over time? Are there clones that are expanding? So you'd assume that if there's a clonal expansion, is that the anti-tumor clone? Kind of what I expected going in. And then what do the clonal dynamics look like over time? Because we had all those serial blood samples. So yes, here's your question. Yes, we did cell-free DNA. So we did the blood cells, ultra diverse, the tumors, very broad range in diversity. Some are very diverse, some are extremely monoclonal. And then the cell-free DNA was even simpler than the tumors. So cell-free DNA has the challenge in that there's not very much of it. So when we're sequencing it, we're only sequencing a very small subpopulation, probably the most frequent TCR clones. But the most exciting finding to us was if you take the tumor TCRs, which we took as most likely to be anti-tumor, and you Venn diagram with the blood cells and the cell-free DNA, the cell-free DNA is 10-fold enriched for CDR3s found in the tumor versus the PBMCs. So you can find them in the PBMCs. The problem is they're all at the same frequency, so it's very hard to pick them out. Whereas the cell-free DNA, there's much fewer of them, but it's much more likely that they're coming from the tumor. We think this is because the T cells that are combating the tumor are turning over, shedding their cell-free DNA into blood so we can actually find them. Cell-free DNA also has a very short half-life, so it only exists in the blood for maybe 16 hours before it's not detectable. So you need a constant proliferative source to be able to find it. That's why it works very well for cancer, but it also works very well for immune response.
Yeah, so yeah, I'm very surprised about this data because the first thing I saw, the cell-free DNA must come from the peripheral blood T cell that are dying. But it seems like for you, that a lot of them come from the tumor infiltrate T cell.
Yeah, because I think the proliferative rate is much, much higher inside of the tumor versus in the bloodstream. It's also surprising in general, if you think about our ability to even find circulating tumor DNA, even the biggest tumor, like maybe is a centimeter or 2. Your whole body is way bigger than that, but 1 to 3% of your bloodstream can have circulating tumor DNA. So it's really more of a functional measure of turning over the cells versus just sheer quantity of cells in your body.
Yeah, excuse me, can I make 2 clarifications because it's all pretty new to me, just for the sake of it, to understand. So first of all, the cell-free DNA and the DNA in blood are the same terms.
Yes, sorry, I should have clarified.
So there
that.
are CF and CT.
Yeah,
Is there any difference?
So this is so cell-free DNA is all the DNA in your blood. ctDNA is the subset coming from a cancer cell. So I use CF DNA because it's not coming from cancer, it's coming from T cells that are within the tumor. But yeah, it's an important distinction. Cell-free is everything, CT is the subset.
Thank you. And the second clarification is for this cell-free DNA to emerge to be out of cell, does the cell have to die?
Yes. Although there have been models that show cell-free DNA can be excreted, but in general, most of the cell-free DNA is coming from a dead cell. The other interesting quirk with cell-free DNA is the cell-free DNA that comes from cancer cells are usually shorter. They undergo a different cell death pathway, and you can actually use that as an additional biomarker, just purely looking at the length of the cell-free DNA, because you get those smaller fragments.
That's fascinating. Can I take 10 more seconds? So does this mean that when you see the curves of this ctDNA dropping, that basically means that there are only the dead cells remaining in the tumor mass and they just shrink gradually.
That's the assumption, but you'd have to do some really fine-grained imaging to really see that real-time shrinkage. There are studies like that actually, where they're doing very frequent blood collections and very frequent imaging to really see what those dynamics look like. Way out of scope for what we could do for a trial like this.
You mean the dynamics course?
A dynamics course of cell-free DNA change versus the what do you want to call it? Like the cell death change. Like that would be a very difficult experiment, at least in a human. Maybe you could do that in a mouse. So we want to understand, looking at the TCR diversity, how do we interpret this data? So the way to read this plot, this is the baseline diversity, which I showed you on the previous slide. And then we went back to the old idea, there's no new ideas, about the change in diversity. And what we found here were really two groups. One group that started low and started cold and stayed cold, so the diversity was low, and it stayed low or got even lower. And there are these patients here which started middling to high and then got even higher. And that was really the lesson here, was your starting point mattered, but also your delta mattered as well. What you wanted to be was hot and get hotter. You could be lukewarm and get hotter. But these ones here were the really intransigent tumors where we're just cold and they stay cold. And it really bore out in the survival analysis as well. If you start cold, stay cold, your survival is very low. Whereas these ones where you could really light up those tumors, those are the patients who are doing very well just by looking at T cell diversity. So diversity is fine, but it tells you nothing about what the TCRs are actually identifying. So that's where we set off to next. And the first finding was no two patients ever rose the same TCRs. Even though they had the same cancer types, they all had different immune responses. So this is all tumors by all tumors. I first saw this plot, I was super excited about this one because it had a huge amount of sharing with another tumor. It was actually the same tumor just done 2 times. So the replicate was great, but it didn't tell us anything biologically. And then we found these other CDRs that were kind of shared, but they didn't match to the base pair. There was really no 2 people, not surprisingly. Immune systems are so diverse, no one rose the exact same 2 TCRs in 2 different people. But we could draw on this new algorithm. So this is actually an algorithm invented by the guy who discovered the T cell receptor, Mark Davis. And his idea is you don't have to raise the exact same TCR right to the base pair. You can tolerate a bit of difference right in the middle where that recognition is happening. So he basically has this algorithm called glyph2, which is essentially will take all your TCR data and cluster it all together. And you can not just cluster it within your cohort. He's published millions of TCRs as a public resource. You can cluster your data with this huge reference set. So we've gotten into this stamp collecting exercise as well. We've built this database called TigerDB. That's the acronym over there. And we've got what we're looking for are TCRs that other groups have solved. So we are interested in 3 groups. The first is CDR3s with validated antigens. These are our favorite. If someone else has seen it, they know what the antigen is. We want to know about it because we can use that as a biomarker. Second are TCRs that have been isolated from tumor cells, but we don't know what the targets are, but at least we know they're T cells that are tumor adjacent, not as good as the top one, but probably recognizing tumor. And then the last one, which is the hugest cohort, just TCRs we've seen in cancer patients. We also have a set of TCRs seen in healthy controls. This is probably the least informative, but it's the easiest data to get, and we put it all into the same database. This database supports bulk sequencing, which is what we built it for originally, but now we've adapted it for single cell. T cell receptor actually has 2 chains, alpha and beta, that come together. Since we're interested in cell-free DNA, these sequences just float singletons, but now with single cell sequencing, you can properly pair them. So this database supports the pairing information. And we support the gamma delta T cells as well as the more well understood alpha beta T cells. So we've got 4,000 of CDR3. So this is a student in my group, Shirin Soleimani. She's read 182 papers, went into their supplemental data, reformatted it all, and put it into the database. So these are the 4,000 most interesting TCRs because we'll call them solved. Most interestingly, a lot of them are against viruses. So this is useful for 2 reasons. One, to subtract the background from viruses we all get, but also trying to understand the role of viruses in anti-tumor immunity, because some of these TCRs are cross-reactive. They may have been raised for a virus, but they're still close enough to a tumor antigen. They're actually informative to understand immune response as well against tumors. We also have tens of thousands of these T cells next to tumors. We don't know anything about them. And we have millions of TCRs from our lab that we've sequenced, and we know nothing about them. But I didn't want to miss the opportunity from study 1. What if we find the same TCR in study 6? We want to make sure that we've got this big repository of knowledge. And then with all this data, you can make these big networks. So inside each of these circles is a cluster of samples that evolve, or of TCRs that have all been found in unique samples. And the color tells you what type of TCR. So the orange ones are the solved ones. We want to look for those ones first. But we also have tumoral converging. Those are found in cancers. And then red are probably the second most interesting. These are the ones that are raised against viruses, but may also be important for antiviral or anti-tumor immunity. So if we zoom in on some of these here for the red ones, all of these are antiviral TCRs, influenza, EBV, CMV. Some of these we see in healthy controls, so we don't really show them. So I put them here just more as a sanity check. Are we finding the viruses we kind of expect? Some of these we are chasing as potential candidates, because some of these TCRs we think are going to actually be useful therapeutically for some tumors. And the other ones that were really the proof in the pudding were these two, mLANA and Team M161. Both of these were already published as antigens or as TCRs that were only found in long-term survivors in lung cancers. So that's what the Mark Davis lab had already discovered. We found the exact same TCRs or TCRs like them in our cohorts, but our cohorts weren't lung cancer. They were breast, ovarian, sarcomas. So in general, some of these TCRs do cut across different cancer types, and same with that mLANA finding. You'll see there's also HLA notation as well. This is your germline variant for presenting the antigens. So not only do they have the exact same TCR, they have the exact same machinery that's presenting the antigen as well. And that's a very important control for trying to interpret TCR data. And this is my last slide. I think we're just coming up to my time. So I hope I've emphasized how important comprehensive profiling of genomics really right from day one. You're going to want to go back to this data set. Every time I've made a panel, I wish there was another gene. It wasn't there. Just go straight for the genome. The survival here was all about the delta. So we really spent a lot of time trying to look at absolute levels, but calibrating each patient individually is very valuable. And then looking at that change to the earliest possible time point to try to understand some of these immune responses. Early expansion, very informative. CD8 T cells, follicular helper T cells. Resistance, very challenging. I highlighted B2M because that was recurrent, but there's actually a whole basket of other immune evasion mechanisms that were scattered across tumors, but you put them all together, those are all the patients who don't respond to immunotherapy. Kind of makes total sense. Evading immune system, activating immune system is not going to do you much good. And then longitudinal specimens and looking for these solved CDR3s. This is totally new. We've just submitted the first paper sharing some of that data. Really trying to understand T cell responses from a T cell specificity perspective. So yeah, this is where we want to go. I've got a whole nother talk on cell-free DNA for early detection, doing comprehensive profiling at diagnosis, and then monitoring over time. Again, using cell-free DNA because you can actually draw out these curves using real data. So yeah, here's my lab. Cindy and Sharon were the two graduate students who really led all this work. The clinical lead with Lillian Sue, and the immunology lead with Pam Ohashi, and the cell-free DNA lead with Scott Bratman. And we patched together a bunch of funding really around all the different questions I put together in this talk. None of this was in one big grant. We kind of parsed it out into different groups. We've published this 3 times, so if you want to read about any of this in detail, those are the 3 papers there. So yeah, happy to take further questions. Yeah, great discussion along the way.
Thank you for the fascinating and thought-provoking